Thank you for tuning into our session on quantifying brain microstructure using multi-parameter MRI. I'm Katie Keenan from the U.S. National Institute of Standards and Technology. In this session, we'll introduce different quantitative MRI methods, their applications, and understand where the measurements and models can go wrong. Historically, radiologists work in qualitative MRI. And now we have powerful quantitative methods available. For this session, we should all get on the same page and start by defining quantitative MRI. So we could define it as anything that has a unit. So this would include distance or volume measurements. Instead, we're going to focus this session on properties of tissue microstructure. For example, T1 and T2 relaxation times, or the apparent diffusion coefficient of water, also called ADC. So we can call all of these images on the right of your screen quantitative MRI maps. And we use these quantitative MRI properties or tools to understand disease or biology prior to any observable changes in distance or volume. So we have this qMRI pipeline, beginning with acquisition and creation of the quantitative MRI maps, and then going into using different biophysical models, such as generating a myelin water fraction map and applying the techniques and the knowledge in either research or the clinic. So I'm going to focus on acquisition and generation of the maps, and subsequent talks will emphasize the biophysical models and application. So rather than having this qualitative image here, we want to get to a quantitative map. In this case, I'll show you an example for isotropic diffusion. So we can probe the water diffusion using the MRI system's gradients. And we acquire a stack of images at different gradient values or different b-value weightings. And then we're going to fit the signal model. So we know that the signal decays in an exponential manner. We have S, which is the signal in these images, and B, the weightings of that we applied, the way we perturbed the system. And then we can calculate the ADC. And so this gets us a quantitative MRI map. In this recent paper by Granzera and others, there's an overview of quantitative MRI methods that are relevant to multiple sclerosis. And here they show diffusion MRI, the example I just showed you, in understanding blood-brain barrier disruption and looking at lesion heterogeneity. Other techniques, such as T1 relaxometry, are used to assess pathology, remyelination, and also used in lesion heterogeneity. So a question that we should ask, and one that's asked in this paper, is can we make these measurements repeatable? Are these methods generalizable to other systems? And I'm going to focus the next part of this talk on T1 measurement. So one of the plenary speakers, Dr. Weisskopf, did a study in 2013 assessing the repeatability of these methods. And a similar study by Grashen et al. Was published last year. So these two studies found minimal bias within a single vendor, a single manufacturer of MRI systems. This is great news that they could get a repeatable measurement, quantitative MRI measurements. It means that the physics can be replicated across systems. Now, in the Weisskopf study, they used the same hardware, the same software, and the same sequence. And in the Grashen study, they used different hardware and software, but it was all from the same manufacturer. And they were able to customize the sequences so that they were exactly the same line by line. So it's very challenging to implement this sort of study at any more than a few sites. So as I mentioned, I'm from the National Institute of Standards and Technology, and we developed a system phantom, or a reference object, in collaboration with the ISMRM. So this object has known values of T1 and T2 water relaxation. We measure those using NMR. And we sent these objects to academic institutions across the United States for T1 measurement. So first, we'll look at inversion recovery, which is considered a reference or a gold standard method. And on this plot, on the x-axis, we have the T1 relaxation time. And then on the y-axis, we have the deviation from the reference or the known T1 value. And you can see that on the whole, there's not much bias from the known value. Here, vendor C is overestimating the T1. And at these last, the last few largest T1 measurements, this is a statistically significant difference. If we look at variable flip angles, this is a faster T1 measurement method. Again, we have the T1 relaxation times on the x-axis, and we have the deviation from the reference or the known value on the y-axis. I want to highlight that I've changed the y-axis limits on you. So on the last plot for inversion recovery, the range of values was about plus or minus 10%. And here you can see that the deviation covers plus or minus 20%. In addition, we can see that vendor B has a substantial bias compared to vendors A and C. And this is statistically significant, again, out here in the longer T1 relaxation times, which is where we have measurements for white matter and gray matter. One question we do get, which is a valid question, is, do these phantom measurements matter? So this study by Lee et al. Did a similar assessment of white matter T1, and so this is a histogram of the measured T1 values across 7 different participants in 2 different vendor systems. So vendor 1 has solid lines, and vendor 2 has these dashed lines. And there's a clear bias in the measurement between these 2 vendor systems. This paper by Stikov et al. Looked at the effect of different measurement methods on white matter T1. So again, we have histograms of T1 measurement, and you can see that using inversion recovery or the reference measurement, and then the variable flip angle overestimates, and the look-locker method underestimates. So more recently, these co-authors worked with the ISMRM Reproducible Research Study Group and conducted a T1 mapping challenge using both the system phantom that I showed you and in vivo data. And they have a great dashboard presenting their results. So I'd encourage you to go look at the differences that they've found when you try to implement T1 measurement across sites without being able to carefully control the protocol. A lot of our measurement methods are changing towards multi-parametric sequences. So how do they perform? This group looked at a multi-parametric method, and here they report measurements in the system phantom that I showed you across 3 different scanners labeled alpha, beta, and gamma. And then they did similar measurements in vivo. And I will say in this case, they saw a greater variation using the phantom than they saw in vivo in healthy volunteers. So there is bias across MRI systems, and it can be established using both reference objects and in participants. I think we're all familiar with the challenges of system upgrades. We had an opportunity to make measurements on a system that underwent a substantial hardware and software upgrade. And we made T1 measurements on this system pre and post upgrade. So I'm going to focus here on the variable flip angle T1 measurement, again, made using the system phantom. And so we have the known T1 times on the x-axis and the deviation from the NMR value on the y-axis. And pre-upgrade, you can see that there is no bias from 0. And we have about a plus or minus 10% range, which is a good measurement, especially compared to what I showed you across vendor systems earlier. Now, post-upgrade, you'll note that there's a shift and we now have a bias to overestimate the T1 measurement regardless of the received coils that we tested. This is a substantial shift. And this site did not identify this issue using their standard qualitative QA quality assurance that they would do on a regular basis. We weren't the only ones to see this effect. So the Lee paper that I mentioned earlier, one system in their study had a software upgrade during their data collection. And so here again, we have T1 on the x-axis, and then it's a histogram of measured T1, and here the black lines are in white matter. The dashed line is a pre-upgrade, and the solid line is a post-upgrade. And we can again see a bias between the pre and post-upgrade data, which is in agreement with the results from our phantom study. So we know that there can be a bias between systems and that MRI systems can change over time. So I use T1 as an example, but I would say at this point, we don't know if there are any QMRI parameters that are invariant to these changes. So if you want to use quantitative MRI parameters as an input to your biophysical model, say to do a myelin water fraction calculation, do you know what the impact is if your system undergoes a software or a hardware upgrade? So what could you do? First, I think it's important to let you know that you're not alone. So there are others in the community that are working on these challenges, and I would point you to the resources from Kiba and Eyeball, and also other researchers at OHBM. My opinion is likely a little biased, but I think it's important to use phantoms or reference objects. I consider them necessary to do site qualification for any multi-site trial, and also for testing throughout the trial, because we don't know when these hardware might start to fail, or you don't always know when the software upgrades are going to take place. Phantoms do add logistical complexity, and the phantom itself may not represent in vivo imaging, but they provide useful information. There are many different phantoms available now. So the ADNI phantom was one of the first widely used phantoms. Over the last decade, we've seen, this is the isotropic diffusion phantom developed with NIST, RSNA, and NIH. This is a phantom for proton density or fat fraction, but it can also be used for R2 star, which is related to iron content. Recently, groups at Berkeley and MGH demonstrated anthropomorphic geometries that could be created using 3D printing and gels. And Walt Schneider is leading an effort to create a diffusion tensor imaging phantom that will be used by many VA sites in the coming years. There are also tools to do standard analysis pipelines, and that'll be discussed more in the next talk. I want to highlight tools to implement the same sequence across platforms. So PulseSeq and HeartVista has software that enable you to sort of bypass the vendor system and run the same sequence out of a MATLAB or Python configuration. I think it's important to say that you shouldn't assume a technique works out of the box. You need to check that when you take these images, you're getting a valid map, and phantoms are key to doing that check. So we learned that if you're able to implement the same sequence across systems, the physics is the same when we go to fit the quantitative model, and you can get reproducible results. The question still, though, is if we can't implement the same sequence, or if we have a hardware or software upgrade, can you take these T1 and T2 quantitative maps and get a repeatable myelin water fraction map, say? So I call on this community to use quality control methods and to include your information about your quality control in image metadata. So thank you for listening to my talk about acquisition and generation of maps. And in the talks that follow in this session, we'll learn about biophysical model and application in research and the clinic. Thank you for your attention.
Hello, everyone, and welcome to my presentations about the benefits and challenges of quantitative MRI. Before jumping into quantitative MRI, let me just remind you how we typically proceed to study the structural organization of the brain with standard T1 weighted images. The aim is obviously to look for differences across groups or changes over time. And to do so, we proceed with voxel-based methods, focusing on the morphometry or the deformation. Those methods rely on several processing steps, going from segmentation of the brain image into its tissue classes of interest, the deformation of these images into some common space, probably some modulation and spatial smoothing, and then finally some statistical analysis, which, for example, here are highlighted by results by Maguire and et al., where they showed a increased hippocampal volume with spatial navigation skills. That said, the interpretation of those morphological differences is not always easy, and these differences, increased in gray matter volume, can be due to morphological differences, like different foldings or increased cortical thickness, but they could also be induced by some problems during the processing at the level of the segmentation or in terms of alignment of the images. Moreover, the segmentation itself, even if it works very well, depends on the image contrast, such that different acquisition sequences, like those two here, will lead to slightly different tissue maps, which here we can observe a different estimate of gray matter volume in these subcortical areas, which of course can be problematic for our analysis. And this is all due to the fact that, as Kathy explained early on, what we are observing obviously depends on the properties of the brain, like their relaxation rates of the various tissues, the proton density, and so on. But it also depends on external technical parameters, such as the scanner hardware and the acquisition parameters. And therefore, the interest of quantitative MRI is to focus on the tissue properties. And this calls for some methods called voxel-based quantification. So here I'll focus mainly on data coming from the MPM protocol, where the Ernst equations are fitted to each and every voxel in order to estimate the parameters of interest, the MR parameters of interest, like the relaxation rates and the magnetization transfer and proton density. In practice, this boils down to acquiring sets of classical images with different weighting, but also acquired at different flip angles and echo times and MT saturations, along with some bias field maps in order to correct for the inhomogeneities in the images. The end results being quantitative maps of these parameters of interest at each and every voxel in the brain. And those images, of course, need to go through a series of processing steps. And because of their nature, some steps need to be a bit more carefully addressed. And I'll focus here on three of them, mainly the segmentation part, the smoothing part, and finally the analytical part. I'll, for the time being, assume that deformations and subject alignment in a reference space will be about the same as we do for VBM analysis. So what are we talking in terms of segmentation? Well, thanks to the quantitative nature of the images, we can observe enhanced gray matter, white matter contrast in the brain. And that will help us greatly with the segmentation of those tissues. Still, depending on the map that we are using, like for example, using the R2 star map, what we observe is that some part of the brain, here the subcortical gray matter, will have an intensity that is quite different from the rest of the gray matter in the brain. That means that in this case, this cortical gray matter should almost be modeled as a different subset of gray matter, as a different tissue class. And because of this enhanced contrast, at least with the empty image, it was proposed to come up with a updated tissue probability maps that do cover more specifically the subcortical areas, where we see here a enhanced gray matter volume to account for the enhanced contrast that we observe in these quantitative or semi-quantitative maps. Others, like Christian, that you will hear afterwards, focused on the brainstem and came up with brainstem specific tissue probability maps. And by combining two types of those quantitative maps, came up with a brainstem specific segmentation that let him more finely study the properties of the brainstem and, as previously mentioned, the difference between groups or their changes over time. So, following this segmentation, assuming that we align our subjects all together, we usually want to apply some smoothing to the data to reduce the remaining intersubject variability in terms of anatomy, but also to reduce the potentially reduce the noise we have in the signal in the images. And let me here just illustrate the problem we're facing with quantitative maps using a 1D example of synthetic data. So here I've generated some signal according to a specific anatomical profile for CSF, gray matter, and white matter, each tissue class having their own specific amplitude. And there is some noise in the data from my 20 subject, as well as some anatomical, a little bit of anatomical variability. If we were to apply on these maps the standard Gaussian smoothing, we would obtain, as expected, a very smooth curve. And that seems all nice, but it isn't. And the main issue comes from here, the interface between two compartments. As you can see, between a pseudo gray matter and pseudo white matter, we have a leakage of the signal, which completely ruins the quantitative nature of the signal that we acquired. And we are mixing pretty much oranges and apples in those voxels. Thankfully, we came up with different ways of smoothing. That is a tissue-weighted smoothing, such that smoothing occur within tissue classes, therefore preserving the quantitative nature within the compartment, like here, gray matter and white matter, and not mixing up the signal between those two. In practice, in 3 dimensions, this is exactly how it works. You would start with a quantitative map in subject space. After warping it into some reference space like the MNI space, and using the associated gray matter probability map, one can specifically smooth the signal within the gray matter compartment. And if it works with gray matter, it also works with white matter, which is another advantage of quantitative MRI, is that we can study both gray and white matter tissues. Now, when it comes to statistical analysis, there are various ways of proceeding. A very simple way is to simply summarize the overall signal per tissue class or region of interest. And here you would look at the signal, the MT value for 3 groups of subjects in the cortical gray matter. And then you can simply do a simple group comparison of these summarized values. And since we have multiple quantitative maps, we can do that across different quantitative maps. And we may also focus beyond cortical gray matter. We can look at deep gray matter or the white matter. In this case, that was healthy controls versus 2 groups of MS patients. Now, obviously, we are typically more interested in proceeding with the standard voxel-based analysis. And here, this is an analysis where we've got results in terms of gray matter, which is here a standard VBM approach. But we can also look at changes in the gray matter for the MT saturation parameter. And then that can also, this analysis can be repeated for the 2 relaxation rates, R1 and R2 star. And overall, those 4 statistical maps can be overlaid in order to present where we observe some significant effect within one or the other statistical maps, and come up maybe with patterns of commonalities in terms of effect, or things that are unique for some of the data that we are considering. And as you can see, this kind of analysis presents 2 sets of issues. First, there is the number of tests that are performed. For each quantitative map, as I mentioned, we can have different tissue classes, gray matter and white matter. So if we go for 4 maps, 2 tissue classes plus VBM, we end up performing 9 tests, which could lead to excessive false positives. And the second part is the interpretation of the results, which can be complex, even though the maps provide complementary information. For example, in this paper by Callaghan and colleagues, they studied the age-related effect, and this simple, or seemingly simple figure contains results for gray matter, white matter, and maps from the MT and R1, as well as the R2 star. And the idea they had was that because MT and R1 are related to myelin and R2 star related to iron, this highlights the places where you can observe a decrease of myelin here in red, mostly in white matter, and where you've got an increase in iron, mostly in gray matter. So given this, it seems obvious that because of the multivariate nature of the data, we should probably go for multivariate statistical approaches. And thankfully, other colleagues, Jigar and et al., came up with an approach called multivariate GLM or MSPM toolbox, where a single statistical inference is performed across the whole brain, for example. But whenever we observe some significant results, illustrated here in these 4 parts of the brain, what we also look at is the profile of the data that drove the observed effect of interest. And in this case, you see a mixture of effects coming from gray matter volume, that is the classical VBM, as well as coming from the quantitative maps that were used as well. And in some places, this is mostly driven by a morphometric effect, while in others, we would more likely observe something that is R2 star dependent, or here that could be a change in proton density and MT saturation. There are obviously other tools flowing around, so I'm just mentioning the few I know of. So in conclusion, as you can intuit, morphometry is still very useful, but qMRI can really help the overall analysis because it enhances specificity. It's also more reproducible across scanners, and potentially it can disentangle disentangle various effects because of the multimodal nature of the data that we are presenting. This allows us to getting closer to this in vivo histology, which allows us to follow tissue changes without or maybe before volume change, and explore more finely the relationships between structure and function in the brain. As I've mentioned, this calls for specific tools for map creation. I'm mentioning here qMRI Lab and the HMRI toolbox, as well as spatial processing, specific spatial processing, and statistical analysis approaches. And with this, I would like to thank the colleagues from the HMRI toolbox team who contributed to those slides, and I thank you for your attention.
My name is Susie Huang. I am a neuroradiologist at Massachusetts General Hospital. Today I'll be speaking about translating quantitative MRI techniques from research to clinical trials and practice, applications for studying neuroinflammation in the brain. And I'll specifically be discussing neuroinflammation in the context of white matter diseases. So what is white matter in the human brain composed of? It consists of tightly packed myelinated and non-myelinated axons, as well as glial cells. And therefore, pathology affecting the white matter primarily involves these components, that is, demyelination, axonal degeneration and loss, and gliosis. So what do we measure with MRI? Within an MRI voxel, which has a spatial resolution of about 1 millimeter or at most sub-millimeter, you can see that there's hundreds to millions of axons and nearly that many glial cells as well. So MRI, in fact, only indirectly captures the effects of these various tissue components on the average signal. And it turns out that the range of available contrast mechanisms with MRI enable us to capture different aspects of the white matter signal composition. Quantitative MRI promises to reveal key features of white matter pathology, and in my talk today I will use as an example multiple sclerosis, which is the most common demyelinating disease of the central nervous system. So the hypothesis and premise of using quantitative MRI is that we can better quantify the extent, type, and spatial distribution and evolution over time of tissue damage in patients with multiple sclerosis using quantitative MRI. And my talk today will focus on various contrast mechanisms through qMRI that enable us to capture different features of MS pathology, including pathology in the normal appearing white matter and gray matter, the heterogeneity and evolution of lesions, brain barrier disruption to some degree, and also this really intriguing prospect of remyelination. I'll begin by discussing the role of T1 relaxometry in assessing disease status and progression. So on the panel here on the left, I'm showing representative clinical contrasts that we typically use to detect lesions and tissue loss over time in diagnostic imaging. So FLAIR, 2D FLAIR, and the 3D space FLAIRs images are very helpful for identifying white matter lesions. Cortical volume can be assessed very well through T1 weighted imaging, such as the MP-RAGE sequence. And more recently, this double inversion recovery sequence allows us to highlight both lesions in the white matter as well as in the gray matter. So it turns out that there are sequences that have been developed that allow us to actually map out the T1 weighted signal evolution, here exemplified through the MP2 RAGE sequence. MP2-RAGE is a 3D MP-RAGE derivative that provides homogeneous T1 weighted images and simultaneous T1 maps that can be calculated from the series of images. And it turns out that T1 relaxometry mapping studies have been used to generate whole brain assessments of multiple sclerosis disease impact and progression. So these studies in the past have found that the global T1 relaxometry changes actually increase with disease progression, predominantly in the later disease stages, and also correlates with brain atrophy. In addition, there's this prospect that being able to quantify T1 throughout the whole brain may provide a personalized approach in which we could assess the heterogeneity of damage in focal lesions, as well as the extent of diffuse pathology by basically quantifying the changes in T1 relaxation times compared to a normal distribution of T1 relaxation times in healthy individuals. So in this paper here, done by Cristina Granciera's group in Lausanne, MS lesions were basically visualized, and T1 relaxation times were quantified within the MS lesions, which were then used to calculate a T1 z-score. And these z-score maps showed that the lesions often appear to be larger than the manually segmented lesions, probably due to areas of increased T1 relaxation times surrounding the MS lesions that were not perceived as lesions per se by expert readers. There are a host of myelin-sensitive metrics that can be derived using quantitative MRI, and these metrics are essential for investigating MS pathophysiology, as it can help us probe lesion evolution and predict outcome, including the effect of therapy. So there's two main quantitative MRI methods for characterizing myelin content, namely quantitative magnetization transfer, or MT for short, and multi-exponential T2 relaxometry. So quantitative MT essentially quantifies the fraction of protons bound to macromolecules like myelin, and it does so by applying selective saturation of protons bound to macromolecules such as myelin, thereby reducing their longitudinal magnetization and rendering it possible to create these magnetization saturation images, or alternatively to be displayed as the ratio of images with magnetization transfer saturation on and off. So these magnetization transfer ratio images, MTR, these are calculated by taking an image with saturation on versus off, and MTSat, is basically an alternative approach that allows us to correct to some degree for the saturation pulses, which can to some degree affect the T1 relaxation times. So in terms of alternative methods to map out myelin, there's also the myelin water fraction that can be derived from multi-exponential T2 relaxometry. So it turns out that T2 relaxometry allows us to be sensitive to different water pools, including myelin water. And this can be basically explored within the central nervous system, primarily in white matter. So both MTR, MTSat, which I haven't shown here, as well as short T2 mapping using multi-exponential T2 relaxometry have been all used to study myelin content and characterize lesions in multiple sclerosis. One of the most important aspects of this work in the past has been actually to validate that these various MRI metrics are sensitive to myelin and specific enough that they may be able to, say, detect areas of demyelination versus remyelination. So focusing first on magnetization transfer, the MTR has been validated by postmortem studies and showed high correlations with myelin when lesions and normal appearing white matter were considered together. Similarly, with using multi-exponential T2 relaxometry, myelin water fraction shows strong correlations with myelin staining in both lesions and the normal appearing white matter, as you can see here on this histological stain, the Luxol fast blue stain, which is binds to myelin. So in the plots below, you can see that there's a strong correlation between myelin water fraction as quantified by multi-exponential T2 relapsometry and LFB optical density, essentially reflecting the myelin content within the tissues, both within averages as well as normal appearing white matter. So using these techniques, which have been supported by histopathological validation, many groups have gone on to assess the prognostic value of MTR and myelin water fraction obtained using these quantitative techniques. The feasibility of measuring dynamic changes in MTR in acute gadolinium enhancing lesions has been demonstrated, as shown in this study here on the left. With acute lesion formation, the average lesional MTR has been shown to initially decrease and then increase. These changes are partially related to edema, but they're also thought to reflect demyelination followed by partial remyelination. The differential effects on MTR recovery in acute lesions have been demonstrated as well for specific therapies, suggesting variations in remyelination capacity. Similarly, myelin water fraction has been studied as a surrogate of myelin content and has been shown to increase over 6 months following the appearance of enhancing lesions, as would be expected in repairing tissue. And so this brings me to, this table here summarizes some of the advantages and limitations to using magnetization transfer imaging and myelin water imaging in clinical trials and clinical practice. Notably, MT imaging is very practical to use. It's implementable on most commercial scanners. It has high precision, but calibration may be required across and within platforms, which is further discussed in Katie Keenan's talk earlier in the symposium. And myelin water imaging has very good intra and intersite reliability and is reproducible, but it has required historically long acquisition times, and there is active research ongoing to try to shorten up those acquisition times. Moving on to other types of quantitative MRI techniques, it turns out that one of the most elusive quantities to measure in multiple sclerosis is axonal damage. So axonal damage is thought to be the substrate of progressive clinical disability, and imaging correlates for axonal damage using MRI have been limited. And these include measures from diffusion tensor imaging, MR spectroscopy. So there's a real need for an improved biomarker of the underlying axonal integrity as a way to assess the therapeutic effect of various neuroprotective drugs that are coming online. So among the markers that have been studied extensively for their sensitivity to axonal integrity include diffusion tensor imaging. Diffusion tensor imaging is sensitive to diffusion both along as well as perpendicular to the axon. It's been very well studied in multiple sclerosis. So quantities like fractional anisotropy are known to be decreased in MS lesions. Mean diffusivity is generally increased. And then axial diffusivity is thought to relate to axonal integrity, whereas the radial diffusivity is thought to be increased in the setting of demyelination. However, various histologic imaging correlation studies have shown that these measures are not entirely specific to axonal integrity. For example, radial diffusivity reflects not only demyelination, but also a component of axonal loss. And this has led to the search for more specific markers of axonal integrity and microstructure using advanced diffusion imaging models and acquisitions. Based on parametric representations of the diffusion MRI signal, a multitude of tissue microstructural parameters can be inferred, such as the relative volume fraction, say, of water within axons in an extraaxonal space, the compartment size of axons, such as average axon diameter, and their orientation distribution. So one advanced diffusion model that has gained traction in the last decade or so is NODI. NODI stands for Neurite Orientation Dispersion and Density Imaging. And this was designed as a clinically feasible multi-B-value protocol that can inform on relative axonal volume fraction independent of fiber crossings and dispersions. So NADI employs a 3-compartment model consisting of neurites, which are essentially axons or dendrites, the extraneurite space, and isotropic diffusion within, say, the cerebrospinal fluid. So the metrics that we can pull out from NADI quantitatively include the intracellular volume fraction, orientation dispersion index, and then the fraction of isotropic water. So NADI has been used in a number of studies in multiple sclerosis, and in general, these studies show that there is overall decrease in the neurite density index within focal lesions. This was shown in the brain by the group at University College London, also corroborated in spinal cord studies. And then in a larger population, the study was published in brain that basically showed and confirmed that there was an overall decrease in intracellular volume fraction within lesions and increase in the isotropic volume fraction. So as a quick summary, in terms of proposed imaging techniques and their role in assessing particularly neuroprotection, DTI is advantageous in the sense that it's very reproducible and has high sensitivity to changes in the tissue, but it's quite limited in its specificity. Whereas advanced diffusion imaging metrics like NADI have gained quite a bit of interest in the last few years for their ability to quantitatively assess axonal metrics, but further validation studies are needed. And in particularly, when we want to consider, say, small diameter axons, even quantifying axonal size, we may need to use higher gradient systems, which are not very widely available at this time. Finally, in terms of other tissue contrasts that can inform about lesion status and particularly the presence of chronic inflammation. So quantitative susceptibility mapping, or QSM, reveals the magnetic properties and has sensitivity to iron and myelin in white matter. So the longitudinal measurements of QSM, the consensus of various studies is that the susceptibility of lesions increases in the initial phase and then essentially normalizes in chronic lesions. And this is thought to reflect the kinetics of iron deposition to some degree in MS pathophysiology, and provides basically a connection between prior histochemical and MR imaging observations of increased iron within lesions as a way to track disease activity over time. So it's known that both myelin loss as well as increased iron content contribute to increased magnetic susceptibility in MS lesions, and this has to some degree created debates about how to interpret increased magnetic susceptibility. A more recent finding has been that some chronic lesions actually exhibit a paramagnetic rim on susceptibility weighted imaging, particularly on the phase and QSM images. But also you can see this on T2 star weighted images and maps. And this persistent phase rim after enhancement resolves. So this was a study that was done at 7 Tesla showing that despite resolution of enhancement within this lesion, there was actually the development of a persistent phase rim, this dark rim around the lesion. And this is thought to correspond to chronic inflammation at the boundaries of the lesion, specifically in reflecting iron content taken up by microglia at the edges of the lesion. This paper by Martina Absinta et al. Basically showed that the persistent phase rim predicted poor outcome in new MS lesions. And so this has led to a very promising way of potentially better understanding and characterizing the heterogeneity of MS lesions and predicting outcome based on the prevalence of such lesions. So in theory, quantitative MRI techniques that measure accurately and reproducibly a biologically specific signal correlating with clinical outcomes would be an ideal candidate for clinical application. However, there are several development steps that are still needed for robust clinical use, and these include the clinical availability of the acquisition methods and tools to reconstruct these parametric maps, methods to extract quantitative information from the parametric maps, and normative values, including cutoffs for normal and pathologic tissue. These sort of quantitative maps also have to be considered in the context of existing diagnostic and prognostic criteria that are used clinically these days. So in summary, in this context, I think quantitative T1 to some degree, the myelin imaging appear to be a little bit more mature and technically advanced and ready for use in studies for using quantitative MRI, whereas the QSM and diffusion-based methods do provide information about microstructure, but may warrant further validation as well as technology development for them to be used in a multi-center setting. So with that, I want to thank you for all your attention, as well as members of my group for contributing to our studies looking at various quantitative MRI techniques in multiple sclerosis. Thank you.
My name is Dr. Christian Lambert, and I'm a consultant neurologist and principal investigator based at the Wellcome Centre for Human Neuroimaging at UCL in London. Today I'm going to be talking about how quantitative MRI can be used to improve subcortical mapping and its application for neurodegenerative disorders. I'm going to divide my talk into 3 parts. In the first, I'm going to focus on the challenges neurodegeneration poses and the current issues with structural brain analyses in this field. In the second, I will try to answer how quantitative MRI can help with this and why it's particularly useful in this field. And then finally, I'll provide a worked example using Parkinson's disease and outline some possible future directions. It cannot be understated the challenge neurodegeneration poses. It is estimated 1 in 3 people alive today will develop some form of neurodegenerative condition, most commonly Alzheimer's disease or Parkinson's disease. The annual cost currently is larger than cancer and heart disease combined, estimated at around 1% of the global GDP, and it is the only leading cause of death whose prevalence is increasing. Importantly, for all these conditions as things stand, there is no cure, no disease modification, and no prevention. One of the reasons for this is that we're simply starting any potential disease-modifying treatments too late, after substantial loss of brain tissue has occurred. By the time people are diagnosed, it is estimated that the diseases have been present for many years, and that there has already been a significant amount of damage to neural tissue. Ideally, we would want to be starting treatments during this period here, known as the preclinical or prodromal period. However, we have no way of accurately diagnosing or monitoring the disease during this period, in the absence of clinical signs. Furthermore, these conditions are highly variable, and it's likely that different subtypes of disease are going to require different treatments. We know that many neurodegenerative conditions begin or have very early changes in brainstem structures and slowly progress over many years before overt clinical signs emerge. Some of the major conditions, such as Parkinson's, are predominantly subcortical diseases, with cortical involvement occurring relatively late during the disease process. As shown here in these lower figures, so Parkinson's is felt to begin in the dorsal motor nucleus of the vagus, before slowly ascending raphe nucleus locus coeruleus and arriving at the substantia nigra at around 10 or 20 years after onset. In Alzheimer's, there's early and severe involvement of the noradrenergic nuclei, the locus coeruleus. Anatomically, the brainstem is a very rich structure, containing well over 180 distinct nuclei that underpin a wide range of critical functions. Despite this, our ability to resolve the internal structure of the brainstem in vivo is limited, as highlighted on the right with the outputs from 2 common processing pipelines. This limited ability to resolve the brainstem poses a significant problem, as highlighted on the next slide. So Parkinson's disease is common, and there's certainly been no shortage of neuroimaging studies in this condition. Parkinson's disease presents as a motor syndrome with slowness of movement in combination with either tremor or excess muscle stiffness, once 60 to 70% of the dopamine-producing neurons found in the substantia nigra have been irreversibly lost. This means the disease-defining hallmark present in every individual with Parkinson's disease is atrophy of this structure. And yet, in a meta-analysis of the VBM studies performed in this condition, containing nearly 1,000 subjects, this finding is notably absent. And this stands in stark contrast to Alzheimer's disease, where they've been very successful and been able to recapitulate the histological hallmarks of this disease, namely the anterior medial temporal lobe and hippocampal atrophy. And the reason underpinning this discrepancy is deceptively simple. Simply put, it boils down to the widespread use of T1-weighted MRI as the de facto structural imaging modality of choice. Now, this provides excellent contrast for cortical structures, which is why Alzheimer's disease has been so successful. And yet in the brainstem, it simply does not have the contrast for many of the structures. And shown here is the output of a Jacobian determinant image, which is the basis for voxel-based morphometry. And what it's showing is that in the brainstem, you simply don't have any information to estimate the volumetric change at a voxel-wise level. And I'll come back to this example later. But simply put, if you cannot map structures at an individual level, you're going to struggle to detect change. Now, one approach that's been suggested as a strategy for this is using canonical regions of interest. However, there's considerable variability within cortical and subcortical structures between individuals and hemispheres, and this will introduce noise that will lower your statistical sensitivity using this approach. Some approaches have suggested normalizing the signal to some reference tissue, or normal reference tissue, creating pseudo-quantitative images. Now, in neurodegeneration, this is a particular challenge because the disease is present in many structures at postmortem, and this introduces a potential confound and bias. For example, pontine tissue disease and Parkinson's disease. So what is it we want from MRI in the field of neurodegeneration? Well, we would like to be able to map the histological hallmarks of disease in life. We want accurate and reproducible biomarkers of disease. We want interpretable measures of tissue damage, and we want to be able to accurately identify these conditions long before the clinical signs emerge. In this second part, I'm going to talk more about how quantitative MRI can aid structural brain analysis and answer why it's useful for neurodegeneration. Now, you will have already heard about many of the positive properties that makes quantitative MRI desirable, namely, it provides more precise measures across scanners and centers, provides greater interpretability at the biophysical level, provides more sensitive measures of pathological progression, improves our ability to map brain anatomy. And in the remainder of the talk, I'm going to concentrate on these latter two features. In some diseases, quantitative MRI can aid traditional visual-based diagnosis at the single subject level by revealing pathology that's hidden on standard T1 weighted MRI. Working through these examples, on the left we have Wilson's disease, which is caused by abnormal copper accumulation within the lentiform nucleus, which you cannot be appreciated on the T1 weighted MRI, but suddenly becomes apparent on susceptibility weighted imaging. In the center example, it's showing dentate iron accumulation across Parkinson's disorders, with healthy controls shown on the left, and then a progressive stepwise increase in dentate iron in Parkinson's disease, with multiple systems atrophy, cerebellar subtype, falling at the extreme end. And finally, in the right example is a rare disease called hereditary diffuse leukoencephalopathy with spheroid bodies. And here it shows the increased white matter damage revealed by the MT imaging that's not as apparent on T1 weighted MRI. What those examples highlight, and the reason quantitative MRI can be used for anatomical mapping, is that it provides a richer characterization of tissue properties and provides more measures to do so, which makes it easier to identify boundaries and regions, which is very similar to the approach taken by histological anatomy mapping. In this example here, we focused on the brainstem, where in the past we used the magnetization transfer and proton density imaging, and used the combined information across both these images to map out and identify new tissue structures and generate new tissue probability maps. And this lower plot shown here reveals why this works. And as you move from grey matter into white matter, you get a progressive increase in myelin density and progressive decrease in proton density as the cellular packing fraction goes up. Having generated these new probability maps, we can now apply these to unseen data as long as we've acquired the same quantitative images. This provides more an accurate mapping of anatomy at the single subject level, which will then improve group level alignment and provide more information about voxel-wise volumetric change and increase our statistical sensitivity. So here's the example I demonstrated earlier showing the Jacobian determinant image from the non-optimized pipelines, contrasted here with the brainstem specific method where suddenly we're capturing a lot of information about regional expansion and contraction at the voxel-wise level. We can combine these analyses with these advances in anatomical mapping with the biophysical insights provided by quantitative MRI measures that allows us to go way beyond voxel-based morphometry and actually begin to understand the basis for our structural brain changes. So in this example, we were examining aging in the brainstem, where it's been described that you get mesencephalic atrophy. What we found was that the regions underpinning this atrophy is volume loss in these structures here. But because we had our quantitative imaging, we knew that in the middle of this volume loss, we were having focal regions of reduced myelin density. And whilst not shown on this figure, we also knew it was associated with increasing proton density. And so combining this information, we were able to conclude that what was happening as people got older was that you were getting focal degeneration of the cerebellothalamic tract. We can build on these ideas still further and use them to improve our anatomical mapping both subcortically, cortically, in the white matter, and also for focal specific regions of interest, including the hypothalamus, locus coeruleus, and cerebellum nuclei. We can also derive and use other quantitative metrics, for example from diffusion or tractography, to provide different ways of characterizing tissue properties, and then call upon exactly the same framework to map structures that are invisible using conventional MRI. And in this example, we really characterize the distributions of similarity in tractography profiles to create feature maps, and then embedded it in the same framework to develop tissue probability maps based off these images, and to segment the thalamus into discrete nuclei that had good correspondence to the ex vivo atlas. Importantly, all of these methods have been developed for 3T MRI. They all can be embedded in the standard processing pipelines and be analyzed using standard techniques such as voxel-based morphometry and voxel-based quantification. And why is this important in neurodegeneration? Well, we know that different diseases have different patterns of subcortical pathology and often target quite discrete focal nuclei. And this may well provide ways to improve early diagnostic accuracy. And I've annotated a few on this figure here. For example, the anterior dorsal nucleus is quite focally affected in Alzheimer's disease, whereas the ventrolateral thalamus is disproportionately affected in progressive supranuclear gaze palsy compared to the other Parkinsonian disorders. In PSP as well, you get atrophy of the subthalamic nucleus, which is not seen in the other Parkinsonian disorders. The locus coeruleus, as described, is a particular region of interest. And interestingly, in the Parkinson's disease and MSA, you seem to find cell loss across the full extent of this structure, whereas in Alzheimer's disease, it's more focal and limited to the rostral middle portion. And then, as mentioned also, the dorsal motor nucleus of the vagus is involved very early on in Parkinson's disease. So in this final section, I'm going to use Parkinson's disease to provide an applied example of these methods, and then briefly touch upon possible future directions in the field of neurodegeneration and quantitative MRI. So we've already seen this slide, but it's just to re-highlight that even with nearly 1,000 subjects worth of T1-weighted MRI data in a meta-analysis, we're still unable to find the focal nigral atrophy that's the disease-defining hallmark of Parkinson's disease. Using the magnetization transfer and proton density imaging in a relatively small sample size of Parkinson's patients and applying our brainstem segmentation method, we were able to apply a voxel-wise analysis in the brainstem and identify the focal nigral atrophy that is the hallmark of this disease, and also capture the increasing iron deposition in the posterior tip, which corresponds with the area of nigrosome 1 that's been found at high field imaging. We can also build on this still further. This slide shows the application of machine learning to the quantitative MRI methods using the optimized subcortical segmentation methods, and demonstrates via learning curves, which is a way of doing power analysis for machine learning, that once you get above 16 subjects, these methods allow you to classify Parkinson's from controls with about an 84% balance accuracy, which is within the error bounds of the nuclear medicine technique known as DAT scanning, which is used clinically. We can also extract the prediction weights from recursive feature selection, and unsurprisingly, they not only resemble the voxel-based morphometry findings, but we'll also start finding additional structures as shown here, which corresponds to the region of the locus coeruleus. In vivo QMRI measures of brain iron are also gaining traction as potential biomarkers in this disease, for example, as a diagnostic marker, so loss of nigrosome 1 in the posterior tip of the nigra, or the regional correlates with disease, motor severity, progression, dyskinesias, or other features such as cognition, which is shown here on the right from some very nice work from Thomas and Weil et al. In 2020. So before I finish, I just want to touch upon future directions in this field. We will have heard from other speakers at this conference, such as Nicholas Weisskopf, how improvements in the biophysical modelling process are providing better mechanistic links to the tissue micropathology. Leveraging these techniques using ultra-high field MRI, so 7T and higher, will allow us to go to even higher resolutions and boost the signal that we're able to gain. There's a lot of active work ongoing developing novel measures which will further improve our profiles of cortical microstructure. And advances in anatomical mapping will allow us to better delineate the variation present at a single subject level, particularly in cytoarchitectural regions of the cortex. We'll expect measurements with improved precision by making them more robust to noise across different conditions, such as motion or across scanners. And finally, taking these techniques to clinical translation to allow earlier, more accurate diagnoses, better subtyping of disease, and more accurate measurements of disease progression. So in conclusion, through this talk we've discussed the challenges for neurodegeneration and the current problems with structural brain analysis, and outlined how quantitative MRI can help, particularly in this field, and finally provide a worked example using Parkinson's disease and outline possible future directions. With that, I'd like to thank you for your attention and also like to acknowledge all the people who've helped support this work over the years through collaborations or funding, or indeed through volunteering in our studies. And I'd now be happy to take any questions you may have in the live session that's just coming up shortly. Thank you.